Powered by Riverside FM.
The cerebral cortex is the brain's outermost layer. It's thin, wrinkled, and grayish in color. This layer contains billions of neurons and plays a pivotal role in our highest mental functions. Folded into ridges called gyri and grooves known as sulci, the cortex maximizes surface areas and neural capacity. It's where perception, planning, movement, and language converge. The cortex is organized into 6 lobes, each with its own specialties. Though each lobe serves unique roles, the cerebral cortex functions as a whole, interconnected, adaptive, and remarkably intelligent. Whether solving problems, forming memories, or experiencing empathy, this layer helps define what it means to be human. The frontal lobe governs decision-making, motor control, and personality. It's where we plan, speak, and express judgment. The parietal lobe processes information like touch, pain, and temperature. It also helps us navigate space and understand our body's orientation. The temporal lobe specializes in hearing, memory, and language comprehension. It's essential for recognizing words and even faces. The occipital lobe, which is located at the back of the brain, handles visual information. It interprets shapes, colors, and motion, and helps make sense of what our eyes are seeing. The insular lobe is hidden deep within the lateral sulcus and is often called the 5th lobe. It plays a key role in interoception, which is our awareness of internal bodily states like hunger, pain, and fatigue. It also contributes to emotional awareness, taste perception, and speech motor control. The limbic lobe, which curves around the inner margin of the cortex, bridges emotion and cognition. It includes structures like the cingulate gyrus and parahippocampal gyrus and is central to memory formation, emotional regulation, and motivated behavior. Microscopically, the cerebral cortex is organized into 6 horizontal or cortical layers, each with distinct cellular compositions and functions. These layers, numbered 1 through 6, from the surface inward, form the structural basis for the cortex's remarkable processing abilities. In layer 1, known as the molecular layer, which is sparse in neurons but rich in dendritic and axonal connections. This contains rare horizontal cells of Kagel and help guide early cortical development. In layer 2, the external granular layer, this is populated by small stellate cells and some pyramidal neurons. It receives input from other cortical areas and refines sensory signals. Layer 3, also known as the external pyramidal layer, is dominated by pyramidal cells, which sends outputs to other cortical regions and support associative processing. Layer 4, the internal granular layer, is rich in stellate cells and receives sensory input from the thalamus and is especially prominent in sensory corticals. Layer 5, the internal pyramidal layer, is home to large pyramidal neurons, including PET cells, in the motor cortex. These project to subcortical structures and drive voluntary movement. Layer 6, the multiform layer, contains a mix of neuron types, including fusiform cells, which send feedback to the thalamus and regulate cortical input. Across these layers, several key neuron types shape cortical function. Pyramidal cells are principal output neurons with long axons. These are found mainly in layers 3 and 5. Stellate or granular cells are star-shaped interneurons that modulate local activity. These are prominent in layers 2 and 5. Fusiform layers are spindle-shaped neurons in layer 6 that connect the cortex to subcortical targets. Horizontal cells of Kagel are rare superficial neurons involved in early cortical wiring. Cells of Martinotti are multipolar interneurons that ascend to layer 1 and inhibit pyramidal cells. Basket cells, whose GABAergic action regulates pyramidal output and synchronizes activity through gamma oscillations linked to attention and memory. Glial cells are non-neural support cells that maintain the brain's structural and chemical environment. They include astrocytes, oligodendrocytes, microglia, and ependymal cells. While glial cells are no longer seen as passive scaffolding, they actively shape neural communication, plasticity, and recovery. Their interconnections with neurons form a tripartite synapsis, where astrocytes modulate neurotransmission alongside pre- and postsynaptic neurons. Cortical regions in the cerebral cortex are broadly divided into evolutionary regions based on phylogenetic development. The archicortex is the oldest cortical structure and is primarily involved with memory formation. It includes regions like the hippocampus and dentate gyrus with a simpler three-layered organization compared to the neocortex. The paleocortex is sometimes grouped with the archicortex under the umbrella term allocortex. This area supports olfactory processing and includes structures like the piriform cortex. Like the archicortex, it typically has 3 layers. The allocortex, encompassing both the archicortex and paleocortex, contrasts with the neocortex by its limited laminar complexity, reflects earlier stages of cortical evolution. The neocortex, also known as the isocortex, is the most recently evolved region, making up around 90% of the human cortex. It's 6-layered, responsible for higher order functions like sensory perception, motor control, language, reasoning, and social cognition. This region is rich in pyramidal cells, interneurons, and columnar organization. Primary areas in the cerebral cortex are the brain's first responders. They receive raw sensory input and initiate motor commands. Secondary areas refine and interpret the raw data from the primary zones. They act as translators, converting raw input into recognizable patterns and coordinated responses. Association areas are the cerebral cortex's integrative hubs. They connect sensory and motor information with memory, emotion, and cognition. Together, these functional areas transform sensation into perception, intention into action, and experience into meaning. They are the architecture behind thought, creativity, and consciousness. When injury strikes the cerebral cortex, either through stroke, trauma, or spinal cord damage, the brain does not simply shut down. It remaps, reorganizes, and rebuilds. This remarkable process is known as cortical plasticity and is the foundation of recovery. An immediate impact of injury, damage disrupts sensory and motor pathways, silencing regions of the cortex. Neurons in affected areas lose synaptic connections, especially in layer 5 pyramidal cells. Functional representations, like hand or wrist control, shrink or vanish from cortical maps. Within weeks, neurons begin forming new immature spines, potentially scaffolds for new recovery. Adjacent or spared regions expand to injured zones, often reshaping motor and sensory maps. Any surviving pathways are strengthened to compensate for lost function. Repetitive movement retrains cortical maps, expanding representation of trained muscles. Compounds like endonerpic malleate enhance AMPA receptor delivery and stabilizes synapses for promoting recovery.